Hello and welcome to my practical on making sense of the eye. Today we are going to dissect a pig's eye in order to get a closer look at the main structures. The equipment you will need to carry out this experiment are a dissecting tray to keep all the parts together, a sharp scalpel to cut through the eye tissue, a pair of sharp tweezers, paper towels to clean up any mess, and finally a pair of latex gloves to stop your hands from getting messy. So let's get started. Here is my pig's eye. One of the first things you'll notice is the extra fat and tissue surrounding the eyeball. In a human, there are 6 muscles attached to the eyeball to enable it to move up, down, left and right. The fatty tissue cushions the eye from the surrounding bone in the eye socket. Usually you would trim this extra tissue off, but there doesn't appear to be a lot on this eye, so I'm just going to leave it. If I turn the eye around, you can see the optic nerve leaving the back of the eye. This nerve carries a lot of the visual information the eye is collecting and sends it straight to the brain to make sense of it. I'm now going to cut the eye in half with a scalpel, straight down the centre, so we have the back of the eye and the front of the eye. You will notice as I'm carefully cutting into the eye with the scalpel, it takes a lot of force to break through the sclera. The sclera is the white part of the eye, covering the outside. The sclera is very thick and made up of many layers of tissue. The clear jelly that is oozing out there is called the vitreous humour and is made up of protein and water. This liquid helps keep the shape of the eye and is clear to let the light through it. Oh, these are a bit better. I have now cut the eye completely in half. If I show you the outside of the back of the eye, you can see the optic nerve again. When you open the inside of the back of the eye, you can see the retina, which is the black lining on the back of the eye that absorbs all of the light entering the eye. I am now showing you the front of the eye again. The hole in the front of the eye is called the pupil. This is where the light enters the eye. The dark circle surrounding the pupil is called the iris. The clear layer covering these two structures is called the cornea. The camera hasn't decided to focus here, but what I'm doing is pulling the remaining vitreous humour from the inside of the eye. Notice that I pull away some of the iris too. I am now gently pulling a structure called the lens from the front of the eye. The lens focuses the light entering your eye. If you look through the lens, everything should be upside down. You can see my hand through the lens but on the opposite side. The lens can be stretched to be made thin to see objects far away, and then go fatter to see objects closer by. The lens is like an onion. It is made up of many layers that grow each year. These layers are made up of protein. Picking up the front of the eye again, you can see the iris, which is a coloured part around the pupil. I'm trying to gently peel the iris away from the front of the eye. The iris is made up of muscle which contracts two ways to allow less or more light in. If someone shone a torch close to your eye, your pupil would suddenly get really small. That is because the iris muscles are decreasing the size of the pupil to let less light into the eye, so it doesn't get damaged. You can probably see the pattern of muscles around the iris. There are tiny lines spreading out from the pupil. What I think of to describe this is the black part of a mushroom. Going back to the front of the eye, an interesting thing to know is that the sclera and the cornea are made out of the same tissue. The difference between the two is that the tissue fibres on the sclera are laid out randomly, so they create an opaque tissue that light cannot get through. And in the cornea, the fibres are laid out in one direction, so light is able to enter the eye through these gaps. If you poke the scalpel through the cornea, you can hear the crunch. This is because the cornea is very thick and protects the front of the eye. This part of the dissection is recorded on my camera and so is very poor quality. You can see the black film called the retina at the back of the eye. This is full of light-sensitive receptor cells that detect the light signal entering the eye. All of the cells send their message to one point at the back of the eye, to the optic nerve. The spot where all these receptors meet is called the blind spot, where no light can be detected. Of course, we do not notice our blind spot, and this is because other receptors make up for the lack of light hitting this one spot. To scrape the retina off, there is an iridescent blue surface called the tapetum. This reflects light back to the retina, but humans do not have this.